Thanks. Thanks a lot for this opportunity to present our work, and I will talk about basically how can we use machine learning to apply for synthetic biology applications for engineering and understanding of microbial systems. And one of the questions we try to solve in our lab from the practical side is to how can we use synthetic biology application to basically accelerate solving the plastic pollution problem. As you know that the plastic is a super versatile material that is very resilient, very light, and due to this super wonderful properties is basically incorporated to every single product of consumer product in our society. And what we try to do, and our long-term goal is to engineer microbial systems enabling to degrade these plastics. And to engineer these microbial systems, basically, you need to express proteins, and you need basically control also metabolism. You need to engineer proteins that will also consume some products of plastics. And for that, of course, you need to make better enzymes that they work faster. And also, if you want to create more complex processes, you need to engineer social life of microbes. For instance, like if one produces enzyme, the other one has to consume and give something back. I will not talk about this part, but mostly focus on gene expression control and actually the protein acceleration. Well, the protein enhancement. So. The first, basically, we started to this idea of okay, in order to make the express proteins, we need to understand how, for instance, what's the information contained along this genotype-phenotype path. We previously found that, for instance, that we can actually gene expression, or protein expression, you can quite well explain metabolism. And that led to idea that we need to quantify every single part in this central dormant genotype-phenotype path. And actually, that's also the other reason why we need to do that is because simply what we are good at the moment is actually is modifying DNA. However, what is actually difficult to understand is how much from the DNA we can, for instance, express of the protein or mRNA. And that's, of course, difficult due to the fact that the genetic code is quite complex. If you look at this, it's basically lots of letters, and that's why we need to use some approaches borrow some approaches from machine learning to understand this code. Let me illustrate you another picture. So basically, here we see every single this distribution. It represents a gene expressed across 3,000 RNA-seq experiment in yeast, and you can see that some genes are can be nearly quite always low expressed, and some genes are quite high expressed. And basically, there is a whole range of expression. And if you think about this picture, that basically this spans everything, like all the experimental conditions, all the technicalities, biological, and the genes are always constrained within a certain range. So these genes are never expressed here, same as that these genes are never expressed there. The other way to see this is a human data, exactly the same phenomenon. So the phenomenon, this is basically also different tissues, different proteins. You can see that some proteins have a bimodal expression, genes have a bimodal distribution, but majority of them basically also constrained within a certain range of expression. And the question was like, okay, if these are basically, there is some sort of constraint of expression, can we actually, is it written in the DNA? Because the only thing what is common across those tissues and is basically DNA. So everything else, conditions, population variation is quite different. And the other way to demonstrate that is you can see that basically within the protein are This is a cross-experimental variation. These two lines represent the fold change. So basically 2/3 of all the experimental variation lies in just a plus-minus fold change. This is a median value of each gene. And this is basically gene expression variation across 3,000 RNA-seq experiment in this case. And you can see that this variation between the genes is way higher than just experimental variation. So, and this is true for over 85% of proteins. So of course there are some of them which are like jumping like crazily between experimental conditions, but majority of the proteins are, or protein expression or gene expression is conserved within a certain range of expression. You can see here just a variance between basically here and this for all the proteins. So on average is 600 times higher the variance between the genes than across the experimental condition. And what the question was, can we just use the DNA information to try to predict the gene expression level from just purely from sequence? And the idea, typically what we do, we have, so Jonathan asked to do it like more informative, what kind of to explain a little bit what kind of techniques and actually how we phrase these problems technically. So basically, we have with data sets that you have letters or DNA expression, well, DNA code, which is encoded as a one hot vector, basically. If it's A, it's 1, 0, 0. If it's G, it's 0, 0, 1, 0, and so on and so forth. And basically, you have these cubes by 4 letters, by sequence length, and a number of genes as a training example. So each of your training examples is a gene within its sequence properties or sequence. And typically, you have, depends how you set up this training, you have from 5,000 to 100,000 examples, and you try to predict Y given the just sequence data, and you divide your data set traditionally by training, tuning, and testing. Tuning you use for validation set, basically for hyperparameter tuning, and this one you only use this at the end of the day. And the way how you evaluate that, you basically compare your prediction to actual experimental observation. And this is a bit of kind of an example of modern, but already kind of outdated. It's an old example, 3 years old, and computer science is quite old. And you might have been familiar with some of these concepts, but I want to just to kind of to exemplify what our modern AI systems can do. And if you get this picture through AI system, it tells you, a group of people shopping at an outdoor market. There are many vegetables and a fruit stand, and this is quite amazing that without anything in any human input, basically this neural network generated this text. And the way how it works, basically, it was done by using 2 different networks. One is convolution, which is good at recognizing different objects, and the other one is recurrent networks, which basically puts these objects as a language model. For instance, if you have like different words like market, people, and they are most likely shopping there, and so on and so forth. And that's why kind of you can make this probabilistic language models. And the idea, you can also apply this in terms of learning rules from or trying to understand what's actually encoded in the DNA. You can use these convolutional networks to identify specific objects, for instance, like transcription factor binding site or other motifs which are statistically popping up there. And basically, then you can put them together in different orientation. And if you do this exercise on basically on a DNA level, try to explain a gene expression or predict gene expression levels, you can see that over 80% of variation can be predicted just from the DNA level. So this is what you give a purely sequence on the x-axis through your neural network. It predicts value, and then you validate that across against experimentally measured values of genes. And each of dots represents one single gene in Saccharomyces cerevisiae in this case. And this is valid for other organisms. We also tested, and of course you can even do that for human with just gene we didn't kind of play around. It has more complex architecture of promoters, like regulatory regions, but there are the reports they can actually do much higher. So nonetheless, basically by learning, like by observing DNA, we can learn and extract some information from it in order to predict the gene expression levels. And then we can ask also the question, what are the most important parts that are responsible for gene expression? Because typically what's done practically, people consider always promoters, that this is the most important thing when you either designing your expression system or interpreting your biology. And basically, if you do this exactly the same exercise, but trying to predict the DNA, you see that typically from each side on alone, there is not much information encoded about gene expression. But if you put this together, at the end of the day, you get this number. And you can try different parts. For instance, yeah, we tried different parts, and you can see that there is a kind of overall the additive interaction across these different regions. So they jointly determine. They jointly determine the expression levels, all these parts. So all the part, there is no single part that is important for gene expression. However, there's a one unit that is important and that is encoding information about gene expression. And what we also tried to do, kind of to make the next step further, like basically abstracting from these black box models by basically trying to understand what is actually encoded in this sequence. And the way how we approach it is very simple. You can imagine, the question is what algorithm recognizes when it looks at the cut picture to understand that if it's a cut or not. Well, basically to make a decision if it's a cut or not. And imagine if you have a perfect classifier which can do this job. The way how you can do that easily is basically to cover a bit of image, like just to put a black patch there, like occluded. And then if you go around with a slicing window across the image, you will find that this is the region where is the most differences between the occluded and non-occluded. And this must be likely the region how algorithm decides if it's a cat or not. And the same, you could do that with DNA and get a score for that particular region if it's important or not. And the way how you can do that, basically, this is just showing for promoter, just to be kind of informative. This is how typical this score looks like for one gene. And you can do that across many genes, and you can basically get this cut beyond what is significant responses, and get these nice motifs, and try to kind of decipher, actually to find some overrepresented motifs that are more important, that we know that has evidence of importance for gene expression. And if you do this exercise, we can find lots of transcription binding sites actually that are non-transcript. So this is a known guy. This is what we basically get from the gene expression, from just this approach with the inclusion, and etc. etc. What I wanted to show here, that basically that there is a unique grammar which is existing across unique motifs which is existing in each of the regions. So that this kind of there is no common letters which are appearing across, for instance, in promoter and terminator will be completely different motifs which are appearing. And then the idea was to see if these motifs are actually can explain gene expression, or observe gene expression levels. And the way how we kind of did it, we tried to kind of quantify if there are basically motifs which are kind of going which are co-occurring as a group of motifs more frequently than any single of them alone, because they found that the alone motifs couldn't explain expression. So we thought, like, if we could do that, that's like more from the perspective of co-occurring groups. And we applied this market basket analysis technique, which is typically used in market analysis. And you can see that basically this is one of the common association that is find in this analysis. If the guy goes to the supermarket, it usually buys diapers and beer together. This is very frequently occurring group of products. And the same idea was to find if the motifs were occurring across the genes. And we found that of course that there are different basically motifs popping up in promoter regions, but generally there are different. There are quite a few significantly co-occurring motifs which are actually present in both together on one gene in terminator and promoter, and or across the different regions. And that was quite cool to see that these are all of them. Remember that they are all important for gene expression. And then to exemplify that, we found this with different rules basically, and to show that the difference, these are genes, and these are all have the same motifs in the promoter region, and depending what they have in a different regions of the gene and the different parts of the gene, the gene expression can vary like by 600-fold. So we can decipher these rules. So based on this kind of logic, we also designed an experiment that actually that we could show that it's not only only the promoter what is important, but depending how you stick to it, which terminator, strong terminator or weak terminator, we can actually regulate gene expression. And these are typically so just will not go to the detail due to the lack of time, but what I want to show, for instance, you have this his promoter, which is if you stick to a different terminator, for instance, strong terminator, you can boost expression by 30-fold of that gene. And this information you can learn directly from the genome. So you don't need to do this kind of like optimization of promoters and so on and so forth. You can learn directly from the genome, essentially from any organism. And that's kind of very important, for instance, when you do the kind of screening for, so typically in practice, how you do gene expression optimization, you screen for native genes, you do some variants, you then verify some mutants, and that's a lot of difficult work. And basically you can source this part, at least that the first two parts, that you can actually learn it directly from the genome, learn the whole regulatory grammar. And that was recently that was done by Jan. Was a excellent work and was recently accepted. So basically, what I want to show again, to conclude that we can actually learn it directly, this grammars by observing gene expression. And now I want to go also very briefly what we do on the protein side. How can we improve the protein optimization? So main challenge in the protein engineering, that typically how it's done, you have a parent molecule, you diversify it, then you try to kind of do the selection, like several rounds of selection to get your desired protein. The challenge, however, here is that there is a lot of, every single introduction of mutation into the protein basically results in loss of function, and you need to do that lots of iteration in order to identify anything that is active. And one way to exemplify it, you can see that number by introducing basically 8 number of mutation fractions of samples that will be active is basically almost the probability is almost 0. And that's why kind of to get this protein diversity practically is very difficult. And I guess if you've seen this basically 2 figures, so this is of course yeah. So this is on the picture on the left is a random sample of phase distribution. So all those people don't exist, and this is how AI makes phases currently. And this is more or less how we kind of design proteins. I mean, proteins are, of course, this is a beautiful picture. Don't get me wrong, it's a Picasso, but you can see still that the cell probably sees that something is unnatural about that, and also that this makes work difficult for it to do. Will not go to the detail what we actually did with here, but basically we applied this approach of generative adversarial networks that can actually learn to generate very complex distribution, but playing this game between the generator and discriminator, basically the generator tries to make fake images that looks like the images from the training set, and until the point the discriminator cannot discriminate them. And then at this point you can use the random vectors basically to map random numbers to actual samples representing from this distribution, and you can do quite cool things from this. Same thing we did basically for kind of proteins. This is just to tell you, this is a quite small network. It still takes a lot of time to train, but the model networks, just to tell you, when you do this kind of exercises, you need to also think about CO2, the way how it's consuming. For instance, this is the latest network built, like it has like 150 billions of parameters, and basically that they take, for instance, this is a life CO2 produced by a lifetime of the car, average car, and this is an amount of CO2 you need to train this network. Just to tell you that this is quite complex experiments. And what we try to do to teach AI how to do this protein, well, not we, but he would teach itself or she. Basically, this is quite complex protein, 300 amino acid long protein, active also in multimeric form, has 2 active sites, and have quite diverse sequence space. And of course, it's quite easy to monitor how you learn what, if this one, for instance, in 2014 faces look like that, and nowadays they look like that. It's easy to get from picture data, but it's difficult to get it from sequence data. So we have to find different metrics how it would work. Basically, what I want to say that we could resemble the natural and so generated sequences would resemble the natural variability existing in sequences. We could find all the active sites. This task would not be possible, for instance, by conventional HMM models, if anyone interested. And to cut story short, I wanted to tell you that basically what's important that we can also, it can learn basically general principles that are existing in a sequence to generate new sequences, and in this way basically generate more sequences that were not present actually in the training data, and very diverse sequences. To exemplify this, please see the right part of this picture. Basically, here you see the number of CAF domains. CAF domains are kind of 3D structural domains that you can recover from 3D structures and assign sequence to it. And the blue line shows the generated diversity of these CAF domains during the training. And this is a control. So if you take your training data and introduce just random mutations that was corresponding to this kind of diversity of these generated sequences, you can see that you are not generating new diversity. But here you can kind of get the diversity. So neural network learns this diversity and introduces new CAF domains that were not existing in training data. We did the experiments, so Martin was doing, and Ella did experiments basically, and we showed that there are very highly active enzymes that are actually, this is to get this randomly, it's virtually impossible. There's a number of mutations existing in the protein. Well, I don't want to go to the details due to lack of time, but the whole point that basically what we find that the important features that protein, when protein looks at the sequence space, that actually make sense biologically. That, for instance, that this so-called attention, where it pays attention, where it discriminates between the good protein and the bad protein, is actually these regions are correlating very nicely with the distance, overall distance, physical distance in protein structures. So just to tell, this was the first kind of generative machine model of proteins, and let's see, soon hopefully will be published. So asking these questions, which were raised by the community. So yes, so what are methodologies? Basically, as I presented, we use the machine learning and kind of different frameworks that are currently popular. Well, what problems? The main problem that sorry, I didn't finish that. The problem that biological sequences are very long, and they have this local kind of information and also global information. So basically, to generate the good sequence, you kind of need to take into account the whole information that exists in different part of the sequence. And you need to have a quite complex modeling approach that can model such distributions. And of course, kind of the way to do that, you need to kind of, when you considering doing these algorithms, you need kind of more understand the data, whether if it's a sequence, for instance, you really need to kind of to get if there is a spatial information there. And the most important kind of what tools you did use to develop yourself. So we had to, there is no like it's an active area of research, basically all kind of tools that we needed to do this for biology had to develop. For instance, one of them was optimizing hyperparameters. In those algorithms, you have crazy lots of hyperparameters, and this basically really depends how, you know, on your performance. You can see that if you, by hyperparameters I mean like learning rates and so on and so forth, that you use for optimization. You can see that basically if you just you know stick to one hyperparameter, you can see that you would never achieve this type of result. And this hyperparameter optimization, this is what takes a lot of time. So this experiment can take up to, you know, several days a week, and that's quite a lot of computation. And also when considering, as I said, sequence data, one has to look into these different kind of mechanisms that can be implemented to kind of to capture this local and global relationship. For instance, when generating these images, this doc image, one has, so this red dot, right? When it generates this red dot, the AI thinks that it also needs to generate the context around it, and this is just for different and the same for sequences. When you're generating a protein, it has to kind of make sense that all the rules of biology is there. Right. So with these words, I want to, of course, thank people from our lab, and also these guys who I accidentally met in some party that actually resulted in eventually resulted in very nice work together, funding, and you for listening. Thank you very much. Yeah, sorry, I took a little bit over. Yeah,
that's okay. Yeah, thank you, Aleksej. But we do have time if anybody has questions. You can either, maybe it's best if you use the raise hand function, or you can also write it in the chat too. But also, excuse me, also, okay, we have one. Anders, you can go ahead and ask your question.
Yeah, thanks. Interesting talk. The classical way of predicting gene expression from sequence data would be looking at the coding part, say this coding adaptation index, and now you indicated there was no information whatsoever in the coding region.
No, I didn't say
of course,
that.
that's reading in triplets and having a table saying which is the preferred codon and so on. Have you tested your method against the classical codon adaptation?
Yeah. So we of course did. So a lot of, I didn't show this information just because of lack of time, but of course. There is a lot of information in the coding region about mRNA expression, and the kind of key finding was there that the same information is also encoded in the regulatory region. So basically, there are these two information, they are overlapping, but they are also additive. And the whole point that when you, for instance, designing genes, right, you cannot just optimize the open reading frame. You need to kind of also take into account how to create all the other parts, including UTRs and things. Of course, and this is definitely important piece of information. And we basically show even experimentally that this is a relevant piece.
Okay. So, but did I misunderstand that the CDS did not contain any information?
No. CDS contained. It was it has a lot of information. CDS has roughly can almost explain half of variation in gene expression.
Okay.
But together they do more. And if you just remove CDS and do only regulatory regions, you can roughly get to the point of CDS.
Okay.
So, but definitely it has information.
Thanks.
Great. Do we have any other questions for Aleksej? I actually have one, maybe a quick one. So, I mean, I think traditionally, at least maybe say 10 years ago, is probably an issue or a challenge with doing this type of analysis in probably obtaining the amount of data that you would need to train these models. Are you finding now that the amount of data is not so much an issue, or is it still a challenge to try and get data sets that are big enough to train these models?
True. I mean, it depends on the kind of on the task. If it's generally in biology, although people will say that we have so much data, but generally in biology we have little data compared to other domains. I mean, from sequencing perspective, yes, we have data in sequence space, but if we're talking about, you know, linking the, for instance, omics data to phenotypes, this is where we kind of still lack a lot of data, and there has to be developments on the technological side that we could, you know, generate data points very quickly and cheaply. We still have, like, you can't, you know, just run a routine RNA-seq experiment with you know, 10,000 or 20,000 of samples. That's not done routinely. So yeah, that's definitely a challenge. But if you work with sequences, I believe you can find some interesting enough information there to train these algorithms.
Okay, nice. All right.
I also have a
case,
question.
sure. Yeah, absolutely.
I was wondering, for the coding regions, did you is it possible to tell apart PCR effects from the biological change or the biological effects? Because
So
PCR.
You mean
So the PCR depends on the how efficient the PCR is depends on the sequence.
Well, yeah. You mean the PCR, the polymerase chain reaction. So basically, what we did, we tried to kind of correct for different biases that existing in the sequencing, like all sorts of GC content and so on and so forth. It didn't explain much of the phenotype. So because there are of course more like, how to say, in all the sequencing, right, you have more biased towards certain nucleotides that will be present in the sequence when you do amplification of your signal, and this didn't at least the GC content and these kind of things, they didn't explain more than, you know, 3% of observed variance. There is, of course, significant, you know, if you do like correlation, but amount of that is super small.
Okay, thanks.
All right. Thanks again, Aleksej.